Dear student, welcome to the part 3 of lecture series of Molecular Beam Epitaxy. In this lecture, we will discuss about the merits of Molecular Beam Epitaxy, the advantages of Molecular Beam Epitaxy, like why the Molecular Beam Epitaxy is the most prominent technique among all the available thin film deposition techniques such as physical vapor deposition techniques like electron beam evaporation and thermal evaporation, and chemical vapor deposition techniques like metal organic CVDs, PECVD, and chemical solution based deposition techniques like sol gel and chemical precipitation, etc. So why this technique is most prominent and very important in microelectronic industry among all available techniques. So we will discuss in this lecture about the advantages, about the merits of molecular beam epitaxy. The first molecular beam epitaxy is that molecular beam epitaxy is low temperature growth process. In this process, the deposited film is basically grown and converted into highly oriented single crystalline and thin film in a directional way at very low temperature. So this technique allows us to grow single phase highly pure oriented thin film along the specific direction at low temperature. Dear student, please note that the range of temperature used in a molecular beam epitaxy is somewhere around 200 degrees centigrade to 800 degrees centigrade. Whereas all the other available techniques like chemical solution deposition techniques or thermal evaporation and electron beam evaporation, the temperature range used is usually more than 800 degrees centigrade. Dear student, question here is that why a material at low temperature is transformed in a high pure way in a single phase using molecular beam epitaxy. Student, please note that the pressure inside the molecular beam epitaxy is ultra high. It is around 10 to the power minus 8 to 10 to the minus 10 torr. So this pressure basically avoids the collision of the molecules, the collision of the precursor molecule and dopant molecule inside the reaction chamber, and they can easily transport it. Secondly, the substrate which is present is also highly pure and directional. So the pressure is basically playing a vital role in order to form the deposited thin film and convert it into a single phase at low temperature. So first and the key advantage of molecular beam epitaxy is low temperature growth, which basically, apart from this, the low temperature play vital role in auto doping. Okay, the purity of material become more prominent in molecular beam epitaxy. Material is more than 99% pure in molecular as compared to the other physical or chemical deposition techniques, because this technique reduce the auto doping. Auto doping is basically an incorporation of impurity from the substrate to the epitaxial layer. Okay, so since the temperature is very low inside the reaction chamber, so the possibility of impurity from the substrate to the deposit or to the epitaxial thin film is less, and due to this we can get highly pure ultra thin film. So this technique basically allow the reduction of auto doping. Another key advantage of this technique is that we basically in this technique we can deposit ultra thin film. So the film is basically very thin, okay. It is in few nanometers, and it is and since you know that the film is usually deposited on the substrate, so the temperature in the inside the reaction chamber is less, and the temperature of the substrate is also less than the incorporation of impurity from the substrate become too less. So this technique basically give us an opportunity to deposit ultra thin film, which is not possible with all the available other chemical vapor or physical vapor deposition techniques. Another molecular beam epitaxy is that this technique allow us to deposit a dopant or to incorporate a dopant in controlled fashion. The dopant introduced in molecular beam epitaxy is in the form of pulse, which is controlled by electronically controlled shutter, which is located at the top of the Epstein cell in which dopant is present. Okay, so this technique allows us to incorporate a precise and controlled dopant into the surface of the thin film or inside the thin film. Dear student, please note that this precise control give us an opportunity to grow the film at molecular level. Okay, and that's the reason that we can get basically an ultra thin film with few nanometer in thickness. Another molecular beam epitaxy is the growth rate. The film grown with the molecular beam epitaxy is quite slow. Usually the film grown in with the MBE is the growth rate of MBE is somewhere around 3000 nanometer per hour, which is quite slow. So due to this, it allows us to control, to manipulate and deposit the thin film at molecular at molecular level, and due to this basically we can prepare our desired material. Please note that the growth is monitor of the film which is deposited on the substrate in the MBE is monitored by read. It's a kind of instrument which is known as reflection high energy electron diffraction. So it's basically a monitor that how the epitaxial layer is grown on the substrate. Another aspect of the slow growth rate is electronically controlled setup or the shutter, which are basically present inside the reaction chamber at the top of dopant container and as well as precursor container. So these are basically act in a pulse fashion, and due to this, the advantage of this technique is that we can basically deposit a thin film also in a layer by layer fashion at molecular level. So this technique basically give us the window of opportunity to prepare or to deposit ultra thin layer of different composition in a layer by layer fashion. Okay, let's say dear student, please note that the MBE is a kind of complex process. Okay, if the chamber of the MBE may contain different types of precursor. Okay, let's say we want to deposit gallium arsenide and gallium aluminium on the arsenide on the substrate of MBE. Usually the substrate is used in MBE is indium phosphate or gallium arsenide. Let's say we want to deposit different type of precursor like gallium arsenide and gallium aluminum arsenide. Initially gallium arsenide from one precursor, if you want to deposit it in a layer by layer fashion, initially gallium arsenide is basically evaporated and deposited in a layer fashion at molecular level on the substrate, and then the shutter basically close the gallium arsenide, and if we want to have another layer of gallium aluminum arsenide, the precursor chamber of gallium aluminum arsenide is basically evaporated, is heated, and the gallium aluminum arsenide is evaporated and transported onto the surface of the deposited gallium aluminum layer, and and we get the gallium aluminum arsenide layer on the gallium arsenide layer. So in this way we can basically prepare a layer by layer epitaxial thin film in a controlled fashion using so this technique basically provide us an opportunity to grow the ultra thin film at molecular level in a controlled fashion, and basically we can manipulate the deposited film at the molecular level using this technique. So this technique is very advantageous. Okay, we can basically get desired properties by depositing desired material at molecular scale. Another advantage of molecular beam epitaxy. Another key advantage of molecular beam epitaxy is that this technique avoids this stagnant layer or boundary layer problems inside the reaction chamber. Dear students, since you know that the pressure inside the reaction chamber is ultra high, okay, pressure inside the reaction chamber is somewhere around 10 raised to power minus 8 to 10 raised to power minus 10 torr, which is quite high. This pressure basically allow the precursor and dopant material to have a longer mean free path inside the reaction chamber, and due to this the precursor and dopant material can be easily transported onto the surface of the substrate. So they cannot be deflected, or they don't deflect somewhere around the boundaries of the reaction chamber. So due to this there is no interaction of the boundaries of the reaction chamber with the molecule, and due to the precursor which are evaporated, and due to this there is no boundary or stagnant layer problem. So no impurities from the boundaries of the reaction chamber can be incorporated inside the reaction chamber, and due to this we can get highly pure material. So this is one of the key advantage of molecular beam epitaxy. If we consider the other evaporation, physical vapor depositions or chemical vapor deposition technique, there is a possibility of boundary value problem, like the incorporation of dopant from the boundaries of the chamber, and they can cause basically defects or impurities inside the structure. However, due to ultra high vapor pressure and controlled precursor precursor and dopant transportation, the boundary problem and the stagnant ion problems are avoided in molecular beam epitaxy. So dear student, thank you for watching this part of the lecture of molecular beam epitaxy. In next lecture, or next part of the lecture of molecular beam epitaxy, we will discuss about the demerits of molecular beam epitaxy, like what are the disadvantages in molecular beam epitaxy. So thank you for watching this lecture.